This is episode 299 of the Stem Cell Podcast, Spinal Cord Development with Dr. James Briscoe. Hello, everyone. This is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. James Briscoe from the Francis Crick Institute. He's on the podcast to talk about his lab's research on molecular and cellular mechanisms of embryonic development, also his experience as the chief editor of Development Journal. We've also got our usual roundup of recent highlights and stem cell news. That's coming up in just a sec.
But first, looking to incorporate neural organoids into your research? Get a free wall chart showing how neural organoids are generated and can be used to better understand brain development. To learn more, visit stemcell.com/brain organoids-wall chart.
You know, Arun, at the ISSCR this year, I'm sure you'll agree, there was an emphasis on translational work. Obviously, it's always translational, but really maybe underscoring the patient experience and the proximity of some of these cell therapies to actual in-human use on a larger scale. I don't want to say a large scale objectively, but expanding, let's say, these trials. But I thought in the talks, when you got more into the basic science, there was a major emphasis also on the fidelity of differentiation, on how you can get these pluripotent stem cell-based systems to approximate the anatomical, molecular minutia, call it, of the actual physiological correlates that they're modeling. And the first story that I have for the roundup is kind of in that vein. This is a story from Barbara Troutline's lab at ETH Zurich over there in Basel, Switzerland, and it's about organoids. Troutline lab has made a big imprint in the field of organoids, and these are neural organoids, which, the great thing about organoids is that they self-organize, and that was another huge component of the last few years of the ISSCR, is the idea that these cells, you put them together and they'll organize into some of the early structures that then can take on more of the complexity of that physiological correlate. In this case, we're talking about neural organoids, which since the beginning have shown remarkable structural and molecular similarities to the primary tissue counterparts, again, that they're trying to model. And in the case of neural embryoid bodies, it's so robust, right? Because, dual SMAD inhibition, you're kind of locking down the system, easier to break it than it is to build. In this case, easier to inhibit. So you get these embryoid bodies that direct towards neuroectoderm, and then you put them in an extrinsic matrix environment, oftentimes matrigel, that allows for polarization and more expansion of the tissue architecture. So specifically, the polarized neuroepithelium around these large luminal regions. And then you get this cell intrinsic extracellular matrix secretion from all the cell types there that can modify the microenvironment. But the dynamics of that are really obscure. We know a lot about the transcriptome and the proteome, a lot of ohms, but the extracellular microenvironment and space, not just in its constitution, I think we're at early stages here. And what we know are from non-human model systems, and of course, brain development, very unique in the human. So that's what this paper is really getting at. How do we observe some of the extracellular matrix and other facets of the architectural morphogenic changes during neural organoid differentiation and development? But it's tough because there's not a lot of methods to dynamically track organoid development. And mind you, the important thing here is that these develop over the course of weeks, months. we heard from Paula Arlotta at the ISSCR just now. She has them going for over 7 years, which is totally insane. She said herself, nobody should try this at home just because, I mean, who has the time, right? But uh, even on shorter time scales, it's really tough. But also in this case, because these organoids are pretty large, right, and they're dense, and the slowness of their development and the requirement to require like a closed system for sterile imaging. It's just tough. It's a lot of things. Um, so in this case, the trial line, they took a pretty straightforward approach. They used these like sparsely labeled, so kind of a mosaic where they threw in a few cells, a minority of cells. They're labeled with two different markers, just constitutive fluorescent proteins, and then they track them and subject them to this kind of image segmentation approach and long-term light sheet microscopy and all kinds of computational tools. And yeah, they didn't go for 7 years, right? But they were able to track these things over 2 weeks of organoid development and at key developmental time points, as the pluripotency gave way to this pseudostratified neuroepithelium, and they saw again this is a straightforward study, but I think the resolution and the elegance of the study and conclusions, I think, is why it was a Nature article. They saw these nuclear migrations, elongation of radial glial cells, and their differentiation to neurons. And then they showed that, and this I think was key and makes you think about all these systems, organoid systems that are incorporating the 3D component. They show that exposure to this extrinsic extracellular matrix, in this case Matrigel, which we've talked about a lot, is pseudo-defined, but a lot of unknowns in there. That it modulates the morphogenesis. It really has a change relative to the intrinsic extracellular matrix that's secreted by the cells. And of course, that tissue pattern, and they showed, is modulation. Is downstream. What is changed? And Arun, I'll give you one guess what signaling pathway this is all mediated by.
It's either Wnt or Yap.
Come on, bro. It's both. We're talking about Wnt by way of Yap. And I guess no one's ever surprised. I mean, it's like a meme on the show, the Wnt. But in this case, the Yap component, it makes sense, talking about size and expansion of tissues. But Wnt does a lot of things, so I'm not poo-pooing it. The fact that it went downstream to Wnt, I think, is kind of a validation. You got to show Wnt, or your system ain't anything. But jokes aside here, the high resolution images, and I invite everyone to have a look at this article and some of the videos. But at the end of the day, I think it introduced a system, right, that's really complex but straightforward. If you can have both in one, this is kind of it. And it gave a mechanistic insight. And for me, the conceptual insight of the extracellular matrix, the balance of the extrinsic and the intrinsic. I think, all of us have seen this in our studies, that depending on what kind of matrix environment, 2D versus 3D, but also, you talk about the Geltrex, you talk about, just basic fibrin or alginate, whatever it is, the matrix environment, not just by way of the structure, but also the signaling, I think is key to faithful development, or just modulating and making differences in differentiation and the architecture of these organoids, because the function is in large part dictated by the structure, not just the molecular biology of these cells.
Good stuff. I mean, there's that movie slash book, Hitchhiker's Guide to the Galaxy, and the question was, "What is the meaning of life?" And the answer was 42. No, I think the actual answer is Wnt signaling. Well, I mean, Wnt does make life, so there's that. But anyways, yeah, really great study here, combining longitudinal live imaging with fluorescent reporters and some basic mechanistic insights into the role of the extracellular matrix here. Troutline Lab has a long history of combining. Yeah, it's one of these like wet dry combo labs, but really has pioneered a lot of these amazing computational approaches in stem cell biology, and I think she received the early like career award or something like that. So it gives you from the ISSCR, so it gives you an idea of the respect that her work has in this particular field, and it's reflected in papers, high profile papers like this. Live longitudinal imaging. I think if if you haven't incorporated it into your workflows, you should. I mean, there's so many fluorescent reporter lines that are out there, and for example, my lab uses a ton from the Allen Institute. We actually had the chance to briefly reconnect with Rugen Wardana, who was a previous guest on the podcast, and she's pioneered a lot of these fluorescent reporters for the Allen Institute for Cell Sciences. But there's a ton of these out there. And why wouldn't you use them? It's a non-invasive analysis. Seeing is believing. You don't have to terminate the populations to get like fixed samples for confocal imaging. So incorporate some of these workflows into your own projects. Use the longitudinal live imaging approach, and I guess we're all still using Matrigel in 2025. I guess there's not many alternatives, or there are alternatives, sir, but Matrigel is still king, right?
Yeah, I guess it's the most straightforward, but I can see it giving way to some of these more defined substrates. But I like what you said about the live imaging, and of course, I mean, it, it is still a bit of a high bar to get yourself, advanced light sheet microscope, but with core facilities and with, the price point coming down, I think if I had a new lab I was starting up, I would make that the foundation of my startup package, as you're alluding to there, because the tools are really amazing out there, and truly it's at the kind of the apex of the original vision of science, right? And particularly biology. We spent all these years making observations, but we can't really see things directly because we're working at the molecular scale, right? And these tiny embryos. But now we've converged on not only being able to resolve optically increasingly small size embryo, not even thinking of, electron microscopy, but the molecular component here with the reporters, as you said, all of those available, and the systems, the model systems, we're getting more and more faithful correlates of real embryonic development, seeing in real time human developmental events, from gastrulation beyond. So exciting, and I think this really indicative, as well as the other story I'm going to come to, maybe two sides of the same coin of how our way of examining the systems and asking the questions is really, through tech and making great strides forward. So I'm glad you raised that point.
Yeah, and as we saw at ISSCR, organoids are just a tool to enable some of this deep analysis of development, but also other disease phenotypes and other uh, observations as well, especially like in the context of, say, primary organoids and cancer. And that's exactly what I'm going to be talking about here. This is a cell stem cell paper coming from Peng Lu over there at the Tsinghua University, which is the basically the MIT of China, if you haven't heard. The title of this paper is An Organoid Co-Culture Model for Probing Systemic Anti-Tumor Immunity in Lung Cancer. And in contrast to some of the developmental studies that we're going to be covering on the show today, this is focused more on primary tumors, organoids created from primary tumors in the context of lung cancer primary cells. Of course, you can make two different types of organoids. You have the iPS or pluripotent stem cell derived, great for studying longitudinal development longitudinally, especially if you're combining some of these new imaging modalities. And forget light sheet. I mean, you don't have to have light sheet to do great imaging. Just get you a basic level, decent epifluorescence microscope. But I think the key, the key, and maybe this is just like me, this is a very personal thing for me because I've been trying to get a light sheet for a long time. You just need a good epifluorescence microscope, Dale. You don't have to go spend those couple millions of dollars. Maybe you have that kind of money, but I don't. But it's the fluorescent reporters. That's what it's all about. And just following up from the previous story. But moving on to this study, this is primary cells, primary tumor organoids, focusing on lung cancer, and This is building on something we just talked about, the role of the microenvironment, in this context, the tumor microenvironment. So in the context of cancer, you got to figure out how the tumor microenvironment and also the immune macro environment, if you think about it, is really important for developing more effective cancer diagnoses, approaches, as well as therapeutic strategies. You got to figure out how the immune system actually interacts with the tumor, not necessarily with the tumor cells themselves, but the greater microenvironment around the tumor, right? So you need good model systems to do that, and they established an exciting co-culture system here using a gel liquid interface or GLI co-culture model of primary lung cancer organoids or LCOs, and they paired these LCOs with peripheral blood mononuclear cells, PBMCs, which we commonly know as the cells that are being used to create iPSCs in reprogramming, but that's not what we're talking about in this context. But the bigger picture is for all of these systems, it's more than just making the organoid. You want to be able to model how that organoid is interacting in the greater context of the body and the greater context of the niche. So these co-culture systems are all the rage, especially in the context of immune. And this actually, a bit of a tangent, kind of relates to some of the work that we're doing in my lab, trying to develop some cardiac co-culture systems, again, to model the role of the immune system in the context of something like myocarditis, right? Because you have inflammation in the heart. It's not just cells directly killing the cardiomyocytes, but you got to figure out what's happening with the immune population. So the dream is to create a cardiac plus immune co-culture system that would help you understand that. And your neighbor, Shubing Chen, has actually done a pretty good job at this. That's my tangent. Coming back on track. This is focusing on lung cancer, not cardiac organoids, but the concept is the same. You want to look at these interactions between the immune cells, the tumor organoids, for optimized simulation of in vivo systemic anti-tumor immunity. So they did what you might expect. They had a huge cohort of lung cancer patients. They demonstrated that these responses in these gel liquid interface models under alpha PD-1 anti-PD-1 treatment reflected the actual real immunotherapy outcomes of the patients. That's awesome, right? You want to be able to model what's happening to the patients in this in vitro setting. And then they took it a little bit further to dissected the different tumor immune processes that are actually mediated by these PBMC derived T cells within these GLI models using a bunch of omics analyses, and the characterization of actually the tumor reactive T cells. So these CD44 positive, CD9 positive T cells with memory like phenotypes. So that's a reflection of actually how effective the immunotherapy was. Um, and I think that's great because it's a model that you want to use to predict how an immunotherapy or how any therapy might do in the clinical setting, in the in vivo setting. I mean, I do the same thing with cardiotoxicity. The dream is predicting clinical phenotypes using your in vitro model systems. And I think this GLI co-culture system might be a unique way to do that in the context of immunotherapy for lung cancer.
Yeah, I think it's a really neat idea. And the dream, I guess, is not just the predictive value, right? And the kind of using cells as an avatar of the patient. Um, but for me, I feel like extending that, so where you see where they're not predictive, if you could then muck around with the model, right, to see if you can endow some adjuvant or whatever, mechanistically understand why it is that it isn't effective in those patients for whom it's not effective, and then use the model to like tweak it to make it effective. I think that's the obvious extension, maybe not obvious extension, or maybe that's not even feasible. But that's what jumps to mind for me is using this not just for patient specific screening of therapies, but also maybe to augment their efficacy. But the other side, I mean, the obvious, of course, extension, I would guess, is that if this is a thing for lung cancer specifically, there's a whole bunch of primary tumor organoids out there. So I feel like, many groups, I'm sure in parallel as well as this, are going after this kind of in vitro uh, immunotherapy in a dish model. It's very exciting and very robust here. Clearly, anytime you can look at patient outcome and then correlate it to your results in a dish, I think that you've reached an endpoint successfully of what we've always talked about is that one of the great use cases of pluripotent stem cells and organoids and primary organoids and cells. So really great study, really exciting clinical implications.
Personalized medicine, baby. Predictive modeling. That's what it's all about. Getting your in vitro cultures to approximate what's going to happen in vivo, like what you're alluding to. And the other thing is, you're totally right. This is plug and play because this is focused on lung cancer, but you can envision how this could be extended to any sort of solid tumor, right? Everything's interacting with the immune environment and the immune system. Every cancer approach, every cancer has some sort of influence from the immune system, I'm sure. And that's why a lot of these are being targeted using immunotherapy. So it kind of is plug and play in that way.
Yeah, and again, underscoring the complexity of the niche interactions, right? In this case, it's a more mainline idea in terms of the immune interaction with the tumor, but they mention it also in terms of the limitations of the studies. Future models should incorporate maybe cancer or tumor-associated fibroblasts. And we've talked in previous roundups how that seems to be the theme, is not just in the case of assembloids, but getting these systems that incorporate multiple tissue types, engineered heart tissue, for example. I think we talked about in the last roundup. And in the meeting at the ISSCR, that was another major component. And the takeaway for me really is that as we move closer to approximating the developmental physiology and anatomy in these organoids, I think what we're realizing is that while the traditional idea, I think we were rooted in this idea, similar to like the Waddington idea of germ layer development, which is true, but I think it confines you to thinking like, okay, you're a cardiac guy, you're within the mesodermal lineage, or neural people, they're within the ectodermal. But like the reality is that these germ layer derivatives are all moving through space in proximity to each other, and they are directing across germ layers the fates, and they are what dictates that underlying architecture in early organogenesis during gastrulation, while those germ layers are literally, just layers of cells. So I think that element of space and time and signaling is being appreciated more and more and being incorporated into our diffs. We're trying to get the signaling environment, not just extrinsically, but actually with the native cells next to each other, and that's helping to recapitulate some of the architecture. But of course, in order to do that, we need a benchmark, right? We need to have a true, ground truth, so to speak, and we're getting there. We've had the single cell, which has been amazing, but now with the spatial, uh, it's going to another level, and that's what this story is. A cell story from Cheng Chi Lin, Southeast Hospital in Nanjing, China, senior contact amongst a very large group. And this was, I don't want to say it's small in scope because it's super ambitious, but it's just E7.5, E7.75, and E8.0 mouse embryos, right? So late egg cylinder gastrula stages, you're starting to get cardiogenesis. Really, the basis of a lot of organogenesis is being laid down in this phase. And so n equals 2 of each of those stages, they did spatial to the limit. What are the numbers here? I think they had 285 slices. They were able to reconstruct 105,000-ish high quality cells that they incorporated into downstream analysis. And the idea here was just, identifying kind of that blueprint, the ground truth in the mouse, of the spatial organization of these primary cell types and their intercellular signaling mechanisms. So they combine stereo-seq, as I said, across those stages. And in order to illustrate the utility of it, they show that there was these cross germ layer signaling networks identified what they're calling as primordium determination zone, or the PDZ, that was at the anterior embryonic extraembryonic interface right in the middle there at E7.75. And Arun, you'll appreciate this. Have a look at the resource because they show that this was critical in the formation of the cardiac primordia. And of course, you can have a look at it because it's available in this web-based portal where you can explore. And you really got to look at this because it really is like a digital embryo and transcriptome. And I don't, and someone's probably already said this, but it's like transcript tomography. You know what I mean? You get these many, many slices. So it's like a CAT scan of the whole transcriptome. And the reason why it's so exciting is because, like everything, it's already like a dinosaur, this tech. I mean, I'm joking, but it's already a bit dated when you consider the kind of resolution and the kind of throughput that some of these spatial systems are incorporating, not to mention the computational power that has been built around them. So I feel like we're moving into this era of, the CT, where the T is transcriptome. I mean, I don't know. I'm going crazy here with my desperate search for acronyms, but I think the reality is truly exciting that not just embryos, for example, or organs on a small scale, organ primordia, but also all of our organoids, embryo models, uh, anything. Doesn't even have to be like a correlate to something physiological, but creating interesting differentiation systems and then subjecting them to this kind of high resolution transcriptomic analysis that enables you to actually see what those intercellular signaling mechanisms and other downstream consequences of that are. I think that we'll be able to glean so much more out of our systems, that will feed forward to optimizing differentiation across the board. So I think this is a really exciting resource just to look at, but also the implications are profound.
Speaking of dinosaurs, I felt like a dinosaur the other day because I did some bulk RNA-seq,
Oh
like,
boy.
era 2010 or something like that.
With that microarray array, Arun? You're using happy metrics?
Yeah, I mean, basically at this point it's equivalent, right? It still gives you great data, of course, but it's nothing at the resolution that you get for this kind of stuff. And um, it's the next level. Yeah, absolutely. Spatial is the thing. And, single cell combined with spatial, all these next gen technologies that we were talking about at ISSCR. Um, what are you doing if you're not getting on that train? And part of the issue is just bringing the cost of these technologies down and making them more accessible to everyone. That's really the dream for me is to, if you're doing organoid work, if you're doing embryology or developmental biology, yeah, I think you have to be able to incorporate some of these single cell and spatial technologies into your workflows because if you're doing bulk on an organoid, then you're losing so much of that resolution and information. So it's just about bringing these technologies to everybody, and there are certainly still limitations of this study too. It's using all these cool technologies, but um, as we know with any digital data set, it depends on what you're feeding into it. And so if you're talking about developmental trajectories, you got to do stuff like knockouts too, just to validate that your trajectories are correct. You got to do a better interrogation in that way. So cool technologies, but for me, it's about at least trying to bring them to everybody.
For sure, for sure. I mean, I'm picturing you there in the lab doing bulk seek on your epifluorescent microscope, just rocking out old school. And I respect that because the truth is, all of this stuff, as you just said, it's all hypothesis generators, right? And it's all just a way of finding the questions that are testable. And the foundation that this kind of tech lays, I think, is really it's actionable, but in and of itself, it's not enough. But in terms of access, I would argue that omics, and maybe not spatial at this point, at this price point, but omics is something that really has been democratized. You talk about bulk seek nowadays. I do my own library preps, and you can multiplex and just send something in at a relatively reasonable price point to the cores that are now ubiquitous, if not commercially available. And I envision that the spatial technology maybe will remain expensive and inaccessible due to the tech involved. But the beautiful thing about this, more than anything to me, is that these resources can be shared, right? And of course, as you said, what you feed in. But as a group, as scientists feed in more data into these digital repositories, I think that the totality of that data set again is going to be profound, especially as we add more dimensions, more species, more developmental stages, and I hate to say it, but the AI, right? Because the AI can weave all this stuff together and again generate hypotheses that then hopefully we'll still need humans around to test.
Yeah, exactly, and that's exactly the point that I was about to make is with all these massive data sets building on top of each other, it would take a million lifetimes for anybody, any mere mortal human, to do this sort of comprehensive analysis. So you got to incorporate AI into this. And the great thing is, as you mentioned, so many of these omics data sets are publicly available. So you just got to get our AI overlords to just tease through them all. And they can probably do it in maybe just one AI lifetime, I suppose. Moving on to the last paper for today's roundup. It is a cell stem cell paper coming from some folks who I'm familiar with. This is a friend of the show, actually, Juan Miller Martin, who we've connected with before, at least on social media, I believe, from Department of Cardiac Surgery, Boston Children's Hospital. Also on this paper is Bill Pooh, who's I've crossed paths with during my time in Boston, just down the road from the Seidman lab where I used to work. Another thing I've noticed here, and this is just completely side note, a lot of the folks here on this paper have Chinese ancestry, and it appears that Cell Press has started to incorporate the Chinese characters into the author names, and perhaps that's a way to make these studies more accessible to folks in China. I thought that was a really cool thing, a nice way to personalize a study and make it maybe more accessible. And I can envision a way that this could be extended to other languages as well, maybe Hindi or whatever. You have the English name, but also the true, so a quote true name in their native language. So that's pretty cool. The title of this paper is Rapid Generation of Functional Vascular Organoids via Simultaneous Transcription Factor Activation of Endothelial and Mural Lineages. And vascular organoids are, of course, a super hot topic. We're incorporating some of these into our own work here in my laboratory because, as everybody talked about at ISSCR, vascularization is one way to take your organoids to the next level, especially if you fuse them with another type of organoid, say cardiac, neural, whatever. So, luminization is one way to do it. So, nutrient delivery to the core of the whatever fused organoid that prevents it from becoming necrotic in the core. So, I think there's a lot of applications for vascular organoids, and people like Joseph Penninger are using them in themselves for modeling various types of diseases like COVID and so on. So they're super valuable tools for studying vascular development, disease, regenerative medicine, a lot of applications. But you got to be able to control the cell types that are found within these organoids, ideally. So controlling the endothelial and the mural cell compartments it's a challenging thing. And so here they're presenting a recipe, a protocol, method, whatever you want to call it, to actually generate vascular organoids from iPSCs via activation, orthogonal activation of transcription factors, and two transcription factors. One in particular that's very well known in this area, which is ETB2, and another one, NKX 3.1, using a DOX inducible or a mod RNA system. So it's controlling the transcription factors that are critical for the specification. And the approach is leading to pretty efficient co-differentiation of iPS-derived endothelial cells and iPS-derived mural cells, which is apparently able to produce functional 3D vascular organoids in only 5 days without any sort of embedding in matrigel or extracellular matrix, which is really cool. After actually exposing the vascular organoids to an extracellular matrix, you get enhanced maturation, as you might expect. You need that niche to take these organoids to the next level. They're able to, in that context, for create even larger and more structured vessels. And then you did the omics. Of course, you got to, just like what we're talking about with organoids and doing deeper analyses, they came in and did the single cell RNA sequencing to figure out what's the heterogeneity of the different vascular populations in the organoids, and the temporal regulation of that transcription factor exposure and expression allowed even further subsophistication into the, uh, as you might expect, arterial, angiogenic, iPS-derived endothelial cell phenotypes. So, and then they wrapped it up. Of course, if you want to sell stem cell papers, sometimes you got to do this. You put it in vivo. They grafted the vascular organoids into immunodeficient mice and were able to integrate with the host vasculature. They formed the perfused vasculature and promoted revascularization actually in models of disease of hind limb ischemia, and also pancreatic islet transplantation. So that's really cool. It's showing that these vascular organoids are quote functional in that way, and I think a ton of papers. I was actually talking to Clive Svensson about this. A ton of papers are doing that last step that was alluded to in this study. It's transplanting whatever organoid into the in vivo system, to show functionality, integration, and potentially maturation of the organoid in the in vivo model. So we're not entirely moving beyond the in vivo system, even though the NIH might want us to do that with this FDA Modernization Act 2.0 and all this other stuff that's promoting the in vitro models. I think there is definitely a value for the in vivo, especially in this context of looking at the maturation and functionality of organoids. So, really cool approach. I think really comprehensive study here, going from the in vitro protocol modification and development to the in vivo application. And just like with the immune system, vascularization in the vascular system is applicable to all different types of disease because we're all connected and we all have blood vessels, right? And blood vessels connect everything, as you're aware.
Vessels are the key, right? Next to every cell. And I had a whole life as a vascular biologist. It was so frustrating, I'll be honest. It was a frustrating life in those times, because endothelial cells from pluripotent stem cells is the challenge. And not for nothing, but this is a bit of a validation. I'll put it differently. This is a validation of some work that I did when I was in Shaheen's lab. I worked specifically with Mike Ginsberg, who they cited here very generously, using a similar approach to make this ETV2 to make these, and Shaheen is still all about it. I guarantee you, Shaheen, if you're listening to this and you read this paper, take a breath, Shaheen. I know maybe you're upset about something. You're always upset about some, but this is good. This is an extension, and I think a really important validation. And I like that they incorporated this other NKX 3.1 for the mural. It's kind of this enforced vascular and mural identity and showing the in vivo function, all that. Great. But fundamentally, my struggle has always been with the endothelial cells. Unlike other cells that we've been able to generate in these kind of directed differentiation systems or whatever reprogramming, is that why is it so hard? And what about the stability? And what about the organ specificity? And a lot of the great thing about this here, again, validated, is showing that these ETB2 cells have the plasticity. They show that they generate all kinds of different endothelial subtypes, and Shaheen will tell you that you can put them in different, I don't want to say any, but a lot of different kinds of systems, and they will cater to the needs of that specific organ, this angiocrine idea, right? So yeah, I love that from an engineering perspective, but what I've always get stuck on is the correlation with physiology. Two things. One is like we talk about how, you know, vascular organoids or vasculature and mural, whatever, is gonna maybe be this the savior, or allow scale, increase of scale of embryoid bodies or organoids, whatever you want to call it. But um, I don't know. From a paracrine standpoint, I think that they can be helpful, but like it's not the cells; it's the perfusion, right? So there is still a fundamental limitation in terms of getting vascularized tissues, in that we need a heart, right? We need the blood. It's not just the angiocrine, although Shaheen will tell you that's the most important thing. But so, by the same token, this idea of adding cells, whereas I wish we could find a way where you get the organ intrinsic vasculogenesis like it happens in vivo, right? The idea that you have these circulating, angiogenic precursors, I think in part may account for some neoangiogenesis. But I do think that in development, I don't think it's a colonization. I think the vasculature is born from within each tissue, from perhaps organ-specific vascular rudiments and precursors. So the idea of incorporating vasculature, I think, whether you're adding it in or growing it from within, I think is something that we need to sort out. Forget about the perfusion, but more along the lines of like, what is more physiological? I feel like most developmental biologists kind of adhere to that one cardinal virtue of the more native, the more physiological the process, the better. And just the mashing together, unlike assembloids, where I feel like you can put two tissues just next to each other. The idea of putting a vascular organoid next to I don't know, neural organoid, and then calling it vascularized, I don't know.
Perfusion, boss, perfusion, Dale. The plumbing needs flow. But that was a pretty bad one. Shane, if you're listening, I apologize. But indeed, actually, it's funny that you brought up perfusion because that's totally what's lacking in this system. There is no perfusion. This is a suspension culture, and as we know, endothelial cells need perfusion. They need shear stress, all this functionality to actually work. Right? It's more than just mashing cells together, like what you're talking about in the vascular plus neural or vascular plus cardiac organoid. You need the functionality to take these things to the next level. There is no perfusion in the suspension culture, which precludes shear stress, which is, of course, a key regulator of endothelial maturation. So that's another way you can make these systems perhaps even better.
Yeah, and, that said, I'm not trying to throw shade, it may sound like it. This still has amazing utility in just understanding all those questions I just kinda addressed, like getting a system where you can test, a lot of different modification. I think that's key, and also there's a lot of elements of vascular disease that truly are cell-based, and not so much about the perfusion or, other components that may be lacking in this system. So, kudos, love it. Love that they cited the paper ETV2 all the way. Shaheen, your stock just went up, boss. Moving on to the interview with a true, well, we're all developmental biologists, but a leader, and someone who I think would adhere to that same cardinal virtue of the native physiology, but maybe not. We'll have to ask. Before we get there, though, a quick message from Stem Cell Technologies. Are you looking for more information on organoids? After this show, you ought to be. You should download Stem Cell Technologies' new ebook on organoid research techniques, developed in collaboration with Wiley Publishing. This essential knowledge briefing details the evolution of organoid technologies from discovery to application, including discussion of key milestones and advances of the technology. A review of key publications and annotated reading lists provide further background on many of the topics covered. Download your copy at www.stemcell.com/organoid ebook. All right, everybody. On today's show, we have with us from the Francis Crick Institute, Dr. James Briscoe. He's group leader there at the Francis Crick Institute, also editor-in-chief of the Development Journal, one of my faves. The Briscoe lab studies the molecular and cellular mechanisms of embryonic development with a particular focus on the spinal cord. In the lab, they use model systems like the chick and also human pluripotent stem cells to develop new computational tools and models to identify the genes involved in spinal cord development and determine how all of those factors work together to organize all the cellular components of the spinal cord. James, thanks again for joining us on the show today.
Thank you for having me. It's a pleasure to be here.
The pleasure is ours. I'm not just kissing up to you because I probably sent in a few articles to development, but I do enjoy the journal, and I've enjoyed your work over the years. You started your lab just around the time that human embryonic stem cells were first derived, and as I said, I personally have followed your work because it has a strong emphasis on the molecular and cellular biology of human development, which has always been my first love and what brought me into the stem cell field. So why don't we start with you sharing your own rendering of the central research focus and goals of your lab's work?
Yeah, sure. So I'm really interested in sort of fundamental questions of developmental biology. So how do you make the right cells in the right place at the right time in the right numbers in a developing tissue? And we address this using a range of model systems. We really focus on the neural tube, the spinal cord, and associated tissues. We take an interdisciplinary approach. So we use sort of classic developmental biology approaches in chick, mouse genetics, but we also have adopted sort of newer techniques as they've come along. So from genomics to bioengineering, computational modeling, and of course using stem cells, differentiating stem cells as a model of developing tissue. So really our goal is to get a picture of development across multiple scales, from molecular mechanism to tissue level properties.
Yeah, and of course, one of those critical parts of unraveling development is figuring out the morphogen gradients and the signaling pathways that make development happen, right? I mean, your work on Sonic Hedgehog has been really foundational in studying neural tube development, and we like to joke here on the show that it's not Sonic Hedgehog, but that everything kind of comes back to Wnt signaling, since it seems like every single paper we cover here on the show has to do with Wnt signaling in some way. But you're focused more on Sonic Hedgehog along with other things. So just kind of broadly about morphogen gradients, how do you think like our understanding of these morphogens has evolved since some of your early studies, and kind of what's next for unraveling Sonic Hedgehog?
Yeah, so I think, morphogen gradients is one of those sort of fundamental developmental biology concepts that's been around for decades and decades. And still one of the prevailing ideas within this field is a concentration gradient and cells reading out their position based on the concentration of that signal to which they're exposed. And a lot of my work, my efforts over the last years has been trying to understand how cells do that. And I think what we found boils down to some sort of simple principles, really. So cells are not passive recipients of that extracellular information, that actively engage with interpreting that information. And that interpretation is via gene regulatory networks, so transcriptional networks that take the morphogen input and convert that into differential gene expression. And we've spent a lot of time trying to understand that, both at the molecular and genomic level, but also building mathematical models, so computational models that allow us to take the data we're gathering in the lab and to combine that into models where we can understand how the cells are converting temporal and spatial gradients into discrete changes in gene expression.
Yeah, I mean, that's an amazing synthesis of the approach, but I think the straightforwardness of it belies the complexity of the challenge, right? And that's what's so amazing. I think most of us in the field would agree with this idea that the more you learn, the more you appreciate how miraculous every embryogenic event is in vivo, because it's so reproducible. You look, it happens the same way I like your assessment there, right? the right cell at the right place, the right signal, right place, right time, and the right number. Because that's the key. It's the multiplicity of all those events happening in perfect lockstep over and over, n equals infinity over the course of nature and biology. And then we try to approximate it in vitro, and we fumble it, right? Because it is so complex, and almost, I think that reproducibility kind of masks that incomprehensible complexity that I'm alluding to here of all these cellular interactions. And part of that, as you described there, is that spatial component, the morphogens, but the timing is the other thing that you described. And that's such a key mediator is, one cell a week apart in development could respond completely differently to the same level of signal.
Or just a few hours apart, in fact.
Just a few hours, right? And I mean, when I'm smacking my head against the wall with my diffs is that these things, you have to sustain the sequence over the course of weeks. And that's what's so tough is all those series of hour deltas in sequence. But your story in Nature, I think, really highlighted the amazing progress we made just a few months back around the new year. But it really emphasized this point of the timing. And it occurred to me in reviewing the work, but as far as we've come, we're still trying to understand and replicate these really fundamental, basic early milestones, as you said, these basic, embryogenic events, vertebrate morphogenesis across the board, but specifically with an interest in human. Can you elaborate on that that Nature story, and how your group and others can leverage this model of human trunk formation?
Yeah, happy to. So, I mean, this really started from an observation that so over the last sort of decade or so, there have been various stem cell models of trunk development. So they're called various things. So gastroloids, trunk-like structures, axoloids. And these are stem cell models that generate neural tissue, mesoderm, and sometimes gut, depending on the particular model. But consistently, they lacked notochord. And that was sort of odd to us because it's a defining feature of all vertebrates, all chordates. And it's sort of an essential part of the trunk of an embryo. So it's the scaffold which supports the elongation of the embryo, and it's also the source of signals for patterning the surrounding tissue. So we were sort of intrigued by this observation. So why do gastroloids, why do these organoid models lack a notochord? So we took a step back and went back to the embryo, and we developed a single cell sequencing map of the early chick embryo during this process of trunk formation. And what we wanted to do was to identify the molecular signatures of the notochord progenitors, and the other progenitors which are producing the trunk. So-called neuromesodermal progenitors, which are generating the neural tissue and the mesodermal tissue. And what we found is that we could identify distinct molecular signatures of these different cell types. And they're closely, so if we look at notochord progenitors and neuromesodermal progenitors, they're closely related both molecularly and in spatial terms. They're right next to each other and a bit intermixed within the caudal region of the embryo. And so that deepened the mystery. So given that they're so close molecularly and spatially in the embryo, why were they missing from gastroloids? So looking at the data we were getting from their single cell mapping, we realized that there was interesting dynamics or implied dynamics of TGF-beta signaling. So looking at genes which respond to nodal and BMP signaling, we could see these changing in the notochord neuromesodermal progenitors and the surrounding cell types. So that gave us a clue to start looking in vitro. So taking human ES cells, we started to look at differentiating human ES cells first in 2D, and looking at the timing of BMP and nodal signaling in particular. And what we found is that if you block all TGF-beta signaling, then the tissue will become, you don't get notochord, instead you get neural and mesodermal tissue, so classic kind of gastroloid cell types. If you let nodal and BMP signaling without any inhibition, so for 48 hours or longer, you end up with gut tissue, so endoderm and lateral plate mesoderm. However, what we notice is this Goldilocks period of signaling. So about 24 hours of TGF-beta signaling, so nodal and BMP signaling. If you just let it signal for 24 hours and then block it, you end up creating a lot of notochord tissue along with mesoderm and some neural tissue. So it's really this exquisite timing of TGF-beta signaling, which appears to be important for notochord formation. So we got that information from those 2D cultures, and then we could extend that into 3D differentiation. So taking aggregates of ES cells and exposing them essentially to the same protocol, which allowed a period of nodal and BMP signaling and then blocking nodal and BMP signaling. And when we did that, we got these organoids with structures that contain notochord in the center, surrounded by mesoderm, and then around the outside was this neuroepithelium. And we could look at in more detail at this. They extended and could be a millimeter or two in length, and were like snake-like in their extension. And looking at the cell types within those, we saw this combination which we're seeing in the elongating trunk, this combination of notochord, mesodermal, and neural tissue together. So that's sort of the broad summary, the overview. But what it also allowed us to do is then look at other aspects of what are the signaling requirements. So we could see that some requirements for YAP and ERK signaling in this process. And we could look at in more detail at the notochord structures we were making in vitro, and could show that using ultrastructure studies, so EM studies, that they had what looked like glycogen deposits, which is a typical feature of notochord, desmosomes, connecting cells, again, a typical feature of notochord, along with extracellular matrix sheath surrounding the notochord-like structures within these in vitro structures.
Yes, so you talk about a number of different technologies there that have enabled the dissection of the early mechanisms of development. You know, you talked about some of these 3D models, EM, and various other in vitro systems as well to tease these apart. And we've been talking about this on the show. I think the hottest subfield of stem cell biology is modeling early human development with these new in vitro systems. But your lab has also really been at the forefront of using single cell genomics to dissect some of these cell fate decisions, especially for neurodevelopment. So, on the computational side of things, what do you think are the most promising computational approaches or technologies to actually integrate these really complex data sets of development into coherent models? What do you think about that?
Yeah, I think exactly. So I think we've been very excited about single cell technology because it gives us for the first time that ability to look at single cell, the transcriptome of developing cells. Of course, one of the limitations of most current technologies is it's a snapshot. You're getting a static picture of the transcriptome. So one of the things we've been focused on is trying to adapt methods, both experimental methods and computational methods, that allow us to get some dynamic information out of single cell transcriptome data. So from on the sort of the on the molecular biology side, on the experimental side, we've been adapting and developing some methods around metabolic labeling that gives us insight into the changes in RNA, not just the static level of RNA at single cell level. And that's adapting some methods that originally Cole Trapnell and Jay Shendor introduced a few years ago. And then to complement that, you actually need new computational approaches to analyze that data. So we've been developing deep learning methods, but using biophysical constraints. So trying to have interpretable deep learning methods that take that dynamic single cell transcriptome data and give us some insight into the trajectories of gene expression as ES cells are differentiating into particular cell types.
Wow. I mean, I'm continuously surprised by how the computational element is integrated into the biology. There's no, it's not just, people like to throw around the machine learning, but it takes a really specific and deliberate programmatic effort, right, to develop these tools and to create these associations. You can't just feed data into the AI, right? You got to train it. What do you think about the direction of science in terms of, the advance toward the in silico media?
Yeah, I think it's very important and very interesting. I mean, this is certainly a huge change I've seen over my career. So when I started as a graduate student, I would spend most of my time at the bench, and 90% of the time doing experiments, pipetting, running gels, and then just a small percentage of my time analyzing the results. Normally, the experiment you did, the answer was clear, it was yes or no. But now when we look at how we do science, actually the percentage of time we're spending doing experiment versus analysis, I think it's completely flipped. So often we're spending 80% of our time doing data analysis. And when we think about that, then biology is really becoming a data science. And we need to think about that in the context of how we're training, how we're learning, how we're developing the field. And I think it really changes our perspective on the future of biology as well.
That's such a great point. Shifting gears. I mean, we're talking about all the tech. We're talking about the new models. But I like having you on the show because you kind of wear multiple hats here, and there's many facets to career in science. We all got a little bit of a side hustle. We got our administrative tasks, of course, mentorship. We love it. All the networking and outreach at meetings. And, Arun and I have this podcast, a lot of new media nowadays, novel ways of communicating the work. There's social media. Arun's very active on social media as well. Not so much myself. But there's real hyper kind of science influencers. I don't know what that is, but it's a thing apparently. But one of the earliest media, the journals, I would argue remain the preeminent platform for dissemination of scientific advances. And the chief editors of those journals toil in obscurity to maintain that steady stream of novelty. So hats off to you, my friend. You've been chief editor at Development since 2018, inheriting the reins from Olivier Porquier, another guest and friend of the show. And you guys are operating in this era of open access and preprints. It's kind of a changing environment. And you're not like Springer, the big journal. I feel like you're like, the underdog. What if any are the major challenges nowadays facing the traditional scientific publishing apparatus? And do you see things evolving more so than they already have as we move forward toward, like, what is your vision that you see it going toward?
Yeah, I think it's a very interesting time to be involved with scientific publishing because, as you say, there's huge amounts of things changing. So from business models, from 20 years ago, everything was traditional subscription-based access to articles, to now increasingly open access is dominating the field, and there's various models of open access business models as well. So on the one side, the whole financial business model side of scientific publishing is changing rapidly. At the same time, over the last 5 to 10 years, we've also seen a change in how we as authors, as scientists, are publishing. So in the life sciences, the rise of preprints is really changing how science interacts with the scientific literature. And I'm a big supporter of preprints, both as an author and as an editor. I think as an author, they're fantastic because they give you control over when you release, you give the world access to your work. It's no longer a journals there gatekeeping access to the work that you've done. And I think with that in mind, I think it actually changes the role of journals, or actually returns journals to the role that they used to have and traditionally have, which is, as an editor, what I want to do every month that the journal comes out, I want to collate the papers in the field that I think developmental biologists should be reading this month. And as a group of editors on development, that's what we like to do. That's what we think our role is to help you, the reader, find the papers outside your immediate field, but those papers within developmental biology we think are going to be important, influential, will inspire other developmental biologists. And I think that role means that community journals like Development have a new potential, a new lease of life, where we can help you navigate the preprint literature by pointing you to the papers that we think are particularly important in the field.
Yeah, it's definitely an evolving landscape right now, but I'm with you. Anything that speeds up the publication process and demystifies it and democratizes it, like open access, I think is ultimately a good thing. And you mentioned how open access is dominating the field, understandably so. But in terms of the technology, of course, something else that's dominating the stem cell field is organoid models. And we talk about organoid models like pretty much every single episode here. Various types of exciting stem cell derived, pluripotent stem cell derived, or primary organoid models. We're also seeing this surge of interest in organoid models to understand human development, like what we've already talked about, right? I mean, in your work, you've used chick embryo, you use stem cell derived systems to study pattern formation. These are complementary systems in vivo and in vitro. But what do you think are the key advantages and limitations of these in vitro systems in particular for recapitulating in vivo processes like those you would actually study in the chick? And how do you think these in vitro models can complement or maybe challenge some of the paradigms that we've already established by studying classic vertebrate embryology, like in the chick, for example?
Yeah, I would turn that bug into a feature. I think one aspect of in vitro models is that they allow you to do the type of experiments, manipulations that you couldn't possibly do in an embryo. So there are experiments you just can't do in an embryo because the embryo wouldn't survive, so you'd have nothing to analyze. Whereas those manipulations you can do in vitro. So you can design experiments that give you more insight into developing processes and then take that back in vivo and ask sharper, specific questions about what's happening in vivo. I think another interesting aspect of stem cell models, in vitro models, is we've already touched on, and that is that the embryo is extremely precise, extremely reliable. It appears to do the same thing every time, get to the same endpoint. Whereas we all know working in vitro, there's much less precision, much less reliability. And that's despite our best efforts, right? We're trying to mimic as much as possible what the embryo is doing, and somehow we're failing, right? We're not doing as good a job as the embryo is doing. So that's telling us that we're missing something. There's things we're not understanding there. So I think there's a real opportunity of using the heterogeneity, the variability of in vitro models to understand what the embryo is doing in order to be reliable and to result in the precise tissue patterning we're seeing in the embryo.
Yes, so these systems are all really quite complementary, but don't sell yourself short, James. I mean, look at these pictures in that Nature article. These truncoids, whatever you call them, there are really a remarkable facsimile of certain aspects, right, of gastrulation and human development, and human being the emphasis on human there. And as you said there, the real key and the feature there, not the bug, is that you can deconstruct all these things and ask all these questions separately to get an answer that has real in vivo relevance. Not to mention, I mean, I hate to bring it back to the therapeutic element, but there is this, the clay for making organs and tissues type stuff. I don't know about organs. That's a bit of a stretch, but the tissues, right? Functional cells is I think one of the major exciting endpoints that we've all been trumpeting since the beginning. I said in the open, I've always admired your work for the focus on fundamental mechanisms, right? But I'm also pretty keen on the implications for therapeutic application. We often highlight on the show here spinal cord injury as embodying that early promise of cell-based therapies, right? After the initial derivation of human embryonic stem cells. You were there. You remember a lot of the stories, a lot of videos you saw was rats regaining motility. Even if it was an animation, you got a standing ovation. Then there was that Geron trial of the oligodendrocyte progenitors that was halted due to the unexpected cystic growth at the site of treatment, clearly linked. I wouldn't say it was a black eye. That's a bit harsh, but I think it was a call. It was like a reality check, right? And it halted that trial. It was a bit of a black eye for Geron, and they kind of got out, right? But in retrospect, I think it was an important recalibration of expectations regarding the pace of clinical advance and experimentation, not just in the spinal cord, but across the board. And amazingly, now here we are, where human trials are, showing efficacy and amazing promise, and everyone's so excited. I think it's a reward for being careful, cautious, and deliberate. But let's be frank here. Partial repair of spinal cords was reproduced. It was a thing. Multiple groups showed this in the end of the aughts. I mean, it was viable. It wasn't like a fraud or anything. And the work has been extended, I would argue, considerably since then. So where are we with spinal cord repair? And what's like the prerequisite to getting back into deploying, I don't want to say human trials, but like at least preclinical trials? Like I don't see it a lot in the news. Where are we with the getting towards spinal cord repair?
Yeah, I think you're right. I think it is an exciting time. I think we've got over that initial flurry of activity, which some, as you said, some of which may have been a bit premature, but certainly had some hints of the potential. It's certainly not something we're working on directly, so I can't tell you anything specific about where we will be in the next year or two. But I think looking at the foundations now we're building from, where we're having a much better understanding of the basics of developing systems, the basics of constructing a spinal cord or an organoid, that's a foundation you want to build from. And I think one exciting aspect of organoids, of course, is not necessarily the direct therapeutic consequences, but the ability to have in vitro models. So if you think about this from a cancer biology perspective, a big development in the 70s was the development of cell culture models of cancer. Now we're all used to these immortalized lines, but they were enormously informative in developing our understanding of cancer, and therefore leading a decade or more later to therapeutic advances. And I think one should think about the organoid field in a similar state. We're sort of now beginning to build from a more solid foundation. And I think the prospects are bright for the future, but it is going to be a long, hard slog. I'm not anticipating huge breakthroughs in the next year or two.
But you're at the perfect place to support that long, hard slog. Of course, you're at the Francis Crick Institute. And as a senior group leader at the Crick, you're deeply involved in fostering the collaborative research that we've talked about here today. So, how do you see these cross-disciplinary partnerships, like between developmental biology, computational modeling, kind of reshaping the future of the stem cell field? And just to wrap things up, tell us more about working at the Crick. What makes it great? What makes it such an awesome place to do science?
Yeah, I mean, I love the Crick. I think it's a fantastic research institute. I think we have the luxury of having core funding, which allows us to get on with our research, without having to apply for grant funding for all we do. Of course, we do apply for grant funding, which substitutes the core, but it means that we have core funding to get on with our work. And that also promotes a collegiate atmosphere where it's very easy to interact and collaborate with other labs, because we have a common resource on which we can draw. And a real strength of the Francis Crick Institute is our core facilities, what we call science technology platforms, or STPs. We have fantastically well-funded, state-of-the-art STPs, which range across the core facilities one would want. And particularly in a broad biomedical research environment, as the Francis Crick Institute is, this gives me, as a group leader focused on a particular question, it gives me access to core facilities that I wouldn't have in a smaller environment. So we have great flow cytometry facilities here because we have a lot of immunologists here. Equally, we have great mass spec facilities and electron microscopy facilities here, and so on and so on. And that's because we're a broad biomedical research institute with core funding allowing us to support those. So I think it's that combination of having a core funding, great facility allows that collegial interaction, that collaboration to develop naturally.
Yeah, I mean, this has been a recurring theme when we talk to leadership in major institutes like, Crick in this case, or IMBA is another example where the core and the technological, specialized technological tools, which are so rapidly developing, they're different. The generation time is, I don't know, 5 years or less on some of these things. But you have to stay really abreast of that tech, and you need to have specialized personnel to operate and stay at the front, in deploying that tech, because it allows you to ask these questions in so much more nuanced ways. As some of the work you talked about today here it illustrates, right? The metabolic labeling to add another dimension
Yeah.
to the snapshot of single cell seq, which itself 10 years ago was only in the hands of some specialized users. So it really is such a key element to again, as Arun was talking about in the context of publishing, but also in terms of science, democratizing the science for someone like, the earlier stage investigators, which we always try to emphasize is really key to the future of stem cell science. And you at that, in your editorial role and your leadership role at the Crick, have really fostered a lot of that young talent. I want to give you credit and hats off.
And can I add one piece to that? Because I completely agree with you. I think supporting the next generation of researchers is essential. And actually, one of the programs we've established at Development is, we call it the PI program, the Pathway to Independence program. And every year we select 8 postdocs who are about to go on the academic job market from applications, and we support them. We support them with some training, some mentoring. So each of the PI fellows is mentored by one of the editors of Development, and we raise their profile by interviewing them in the journal. And then we follow their trajectory as they go on the academic job market and see if and when they get their academic positions. So that's a little thing that Development can do as a community not-for-profit journal to support the next generation of researchers.
Yeah, and it moves the needle. I'm aware of that piece. I've followed it because you see familiar faces sometimes that you're aware that you see in papers or whatever. And it really does help to raise the profile. And that's another thing that we really hope to emphasize in the show. So thanks for mentioning that. And thanks for all the work across the board you do to that end. Before we let you go, we got a couple of science peripheral questions we like to ask. In your case, the first is if you were not a scientist and journal editor, what do you think you'd be doing with yourself?
Well, I guess it's too late for a professional tennis player, I suppose. I mean, I've always thought, setting aside my lack of natural talent and, the amount of unrelenting training one would have to do, it particularly appeals because you would live in eternal summer, right? Every professional tournament is in the summer, wherever it is. But I think it's maybe too late for that career choice. I think if it wasn't that, I think, one of the things that fascinates me about developmental biology is how you turn two-dimensional objects into three dimensions. So I think, something like architect or engineer would be an interesting profession. I also enjoy making clothes and sewing, which is also about turning two-dimensional fabric into items that fit a 3D body. So maybe I should have been a tailor or dressmaker or something like that.
Wow, what a neat collection of unrealized dreams.
That's
I think
right.
it would be
My next
great
life,
at
my next career, yeah.
Finally, what's the biggest misconception about science that you'd like to resolve?
Um, yeah, good question. I think a particular misconception that I'd love to resolve is sort of this myth that science is about lone geniuses having sudden eureka moments. I think, we all know the reality of science is about persistence and collaboration, and progress only comes from countless hours of hard work and failures and gradual building up of piece by piece of understanding. So I think you've got to have resilience, and you need to learn to bounce back from failure and do that and be able to collaborate. So I think it's that misconception, because I think it's an important misconception that we need to resolve, because I think it discourages a lot of people from pursuing science initially, because they think they have to be naturally brilliant rather than just curious and persistent. And there's I think related to this, there's I think it's an Isaac Asimov quote that I really love, which is, the quote goes along the lines of that the most exciting phrase in science, and the phrase that normally heralds discovery, is not eureka, it's this looks a bit funny.
Hmm.
And I think that's an important thing to remember, because okay, so getting a funny result normally is because you've added the buffers in the wrong order, or you've got a bracket in the wrong place in your code. But just sometimes it is something you weren't expecting to happen. It is something that you should keep your eye on, watch out for again, because it may be telling you something.
Yes. Oh, that's such a good one. I'm glad to be reminded, because I remember looking through for, I don't know, some speech I had to give. I had this book of quotations, and you look at the Asimov, get to Asimov, and he's got all these
Yeah.
great, any technology advanced enough is indistinguishable from magic, all these ones that are so uh, prescient about science and the role of science in advance. But those are the things that you really feel as a scientist, both on both sides, as you just alluded to. This idea of that you think you're on the right track and you're totally wrong, and it takes two to think and someone to reflect the result off, to be like, but I don't know, this doesn't look right. But also, even better, is that serendipitous, so in the best of times, we all know what that is, and reflect on the joy of it. And thanks for sharing not just that, but all that you've talked about. I think you have a particular insight in the path you've taken as a scientist and thinker. And I think our listeners are really going to enjoy it. So thanks for sharing.
Thank you very much. That was a lot of fun. Thanks for having me.
All right, guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCellPodcast.com, or via email at info@STEMCellPodcast.com. Feedback or to suggest guests. All right, you guys, thank you so much for listening. Until the next time, we're out.